Today we are going to sort of continue talking about VDJ recombination as we talk about B-cell development. And really, the two are very closely related because VDJ recombination is happening in B cells as they develop. And so in some ways, you can think of B-cell development as being the wheres and the whens for VDJ recombination. So recall that this whole process is happening in B cells, and our activated B cells are eventually going to secrete antibodies. So in order to make the antibody that we are hoping to make through this generation of diversity, this process is going to need to occur in some activated B cell. Notice also that our B cell has a receptor on its surface. It is creatively called the B cell receptor. And that receptor has the same specificity as the antibody that is on this, that will later be secreted by that B cell. We talked a little bit about how this happens last time. So after we do our VDJ recombination, in order to make that highly specific variable region for our heavy chain, as well as our VJ recombination to get our highly specific light chain, we will have transcription and translation. Alternative splicing will allow either a hydrophobic segment and this transmembrane domain, which you can see on the left. I will tell you, just to avoid confusion, figure 3.9 and figure 6.17 are in opposite orders. So this protein corresponds to this transcript. So you have to cross over. Or we can have a different kind of splicing where we don't have that hydrophobic segment, we don't have that transmembrane domain, just shown here on the left in the transcript, thus allowing for our secreted antibody to be formed. A lot of what you're going to see with B-cell development has to do with the production of the B-cell receptor, but that B-cell receptor is being made by VDJ recombination. So really, this is still that same VDJ recombination antibody production process. We're mostly going to be calling it the B-cell receptor today, more so than the antibody, but that's because really they're one and the same. And so you can see that here again, where we're largely here going to talk about this B-cell receptor as we talk about B-cell development. It is going to be one and the same as the antibody, but usually we think a lot more about the receptor with developmental stages. So when we think about developmental stages, there are a few things that we can say. So we've already mentioned this process of hematopoiesis, where we have blood cells developing from some type of hematopoietic stem cell. Our B cells come from that hematopoietic stem cell via the common lymphoid precursor. You may see the CLP on a couple slides later today. It's just the lymphoid precursor. And so this is really the origin of cells that are going to go through B-cell development. As we mentioned before, hematopoiesis largely happens in the bone marrow. It will happen a bit in other places like the yolk sac and the fetal liver before birth, but after birth, your hematopoiesis is all coming from bone marrow. I have one other thing I want to say about hematopoiesis in general before we zoom into the B-cell part of hematopoiesis. So if you were to do bone marrow transplantation, you would in fact restore the ability of an animal or a person to perform hematopoiesis. So we can get rid of immune cells in an animal with radiation. We're getting rid of rapidly dividing cells, and most of your cells of your immune system are rapidly dividing. You could also imagine maybe a person with immunodeficiency who is missing an immune system, is missing some of these cells. You could imagine a person who undergoes radiation as, say, cancer therapy. We can transfer bone marrow in and restore hematopoiesis. Typically, when we're doing this process, that bone marrow that we transfer has a mixture of cells in it, a small number of which are the stem cells. Your textbook has a really nice, it goes through a similar experiment to this, talking about how stem cells were originally isolated and things like that. So we can do some stem cell isolation. We have much more sophisticated methods of stem cell isolation now. I'm not going to focus so much on that, but yes, we can do stem cell isolation. One thing that is not well depicted on this figure is here it looks like we are injecting the mouse in the middle of the back. In fact, when you do a bone marrow transplant on a mouse, you actually do an intravenous inoculation into the tail vein. So typically this is going in IV in the tail vein, which is going to be important on the next slide. And those tail vein inoculations are slightly technically challenging. You need to do a fair amount of practice before you are really good at hitting tail veins frequently on a mouse. I don't even know how many mice I had to practice on before I started doing bone marrow transplants. The reason why I tell you this is because of the data presented in this experiment from an earlier edition of your textbook. Here, scientists isolated stem cells specifically and tried to deliver or transplant just those stem cells into a mouse. And their question was, how many stem cells does it take? How many stem cells do you need to get a whole immune system developing from them? So first they transferred 20 stem cells, and 100% of the mice had a full immune system reconstituted. They went down to 10, it was about 90%, 5 about 50%, 2 23%, 1 about 21%. So even transfer of one stem cell is enough to rebuild and reconstitute an entire immune system. So these stem cells have a lot of potential. My guess is that some of these numbers, especially the number for one stem cell, is lower than it could be, because my guess is that some of this, when they did 41 mice, is that they missed the injection. Doing an injection of one cell would not be the easiest thing ever. And so my guess is actually it works a higher percentage of the time than this, but this takes into account how frequently they miss the injection, because it is a tricky injection, especially when you're getting down to so few cells. With the 20 cells, you could miss the injection, only get 10 cells in, and that could be enough. With one, if you miss, you're kind of out of luck. So this, I hope you can see, tells us that these stem cells are incredibly potent and can give an entire immune system, not just B cells, though today we're going to watch what happens in terms of B cell development. Hematopoiesis, of course, happens in primary lymphoid organs. All of these stem cells are coming out of our primary lymphoid organs, and our B cell development is all taking place in the bone marrow. So we'll be focusing on bone marrow today. In fact, all of the steps of our B cell development, going from that hematopoietic stem cell until we have a mature B cell, are happening completely within the bone marrow. Here you can see a bone that looks like I drew it, where all B cell development, as well as this process of selection that we'll see today and on Monday, are happening. This is entirely antigen independent. So we don't have the B cell seeing antigen. We can come back to what that exactly means when we get to the end of selection. There's sort of some antigens at the end of selection, but we really think of this as the antigen independent phase, and our B cell starts to see antigen after it leaves the bone marrow and finishes all the steps we're going to see here. You are making about 5 million B cells a day coming out of your bone marrow. So, and one thing I want you to notice is that the 5 million number is written here at the end 5 million B cells are making it out of your bone marrow per day. 5 million B cells are doing this whole process successfully. You'll see that there are places where the cell can fail throughout this process, and so way more than 5 million cells are starting because many of them are going to fail. The first question or the other thing that I want to just remind you, I've told you this before, is that this process is happening in the absence of antigen. You may be getting sick of me telling you this by now, but as I've mentioned a couple times, this is by far one of the most common misconceptions people have with immunology, and so I want to make it really clear, make sure you're seeing it as frequently as possible so we can try to clear up this process. So as you can see, our generation of diversity is really happening without antigen. We see antigen only later when the B cell is circulating throughout the body. So here is, again, that beginning of this process. This process of B cell development is happening in the bone marrow. And the first question that you might have is, why bone marrow? What is special about the environment inside the bone? What is special in the bone marrow that allows for these early steps of hematopoiesis of all cell types, and particularly all of our steps of B cell development? The important thing to know about bone marrow is that your bone marrow has a number of different structural cells inside it. We usually refer to those structural cells in immunology as stromal cells that are able to interact with our developing B cells. You can see, for example, a cell that's interacting with a developing B cell here that may provide different types of cytokines or other factors to help that B cell along. So these B cells really need to be interacting with lots of stromal cells in the bone marrow in order to receive the signals they need for development. As we think about B cell development, and this will be true of T cell development as well, you can think of our developing B cells and T cells as being really, really, really needy. These cells frequently need a little pat on the head that says, "You're doing a good job. Good job, good job. Stay alive." And if those cells do not frequently receive these types of signals, the cells will undergo apoptosis. And so all of these interactions with stromal cells are giving our developing B cells that support signal to help them stay alive throughout the early stages of this process. This figure shows a lot of different types of molecules that may be interacting with our developing B cell from our stromal cell. The only one that I'm going to mention specifically by name is a cytokine known as IL-7. IL-7 is one of the most important cytokines for long-term survival of lymphocytes, and it's really, really, really key in lymphocyte development. So all of our developing B cells will have an IL-7 receptor. The receptor structure is shown here. It's something that we'll deal with much later. IL-7 is secreted by the stromal cells. It interacts with the IL-7 receptor and signals on our developing B cell to allow things like survival, proliferation, and different B cell specific types of events. And so this process is incredibly important for keeping our developing B cell alive at the beginning. A couple of you had some questions for me when we were doing the flow cytometry experiment where I had a problem that involved something called CD127. CD127 is actually the IL-7 receptor alpha. You didn't need to know that for the problem, but FYI, that's what it is. So bone marrow is important to give our B cell survival signals. And throughout this process, you're going to see our B cell needing that pat on the head for some survival signals. Yes, Molly?
I don't want to muddy the waters between B cells, but
Yep.
is IL-7 just important for B cells or is it T cells?
It is important for developing T cells as well. And it's also important for long-term memory cells to be able to live for a long time. It's kind of the live cytokine, the survive cytokine for a lot of our lymphocytes. So our B cell is going to go through a few different phases. Those that are in yellow are the phases that happen in the primary lymphoid organ. Those that are in pink happen in the secondary lymphoid organ. And so right now we're going to see our diversification process, where we're going to actually make the full diversity. Usually we talk about making the repertoire, or generating a repertoire here. So we're going to generate our repertoire, and then we're going to actually do testing of those B cells, all within the bone marrow. Then only later will we actually start to deal with infection in those secondary lymphoid organs. I told you before that our B cell needs some positive signals to tell it to live. And so before we can talk about all of the details of B cell development and how the B cell performs these early phases, we have to think a tiny bit about how the B cell receptor signals. Because you need to know what the receptor is that's going to get these pat on the head signals. You need to know a few things about this receptor. So we need to think about activating our B cell. That B cell is going to largely be activated through most steps that we see through the B cell receptor, which we've seen before. That B cell receptor, of course, undergoes this nice fancy VDJ recombination process. It has great specificity. It's really just like an antibody. But before we say anything more about the B cell receptor, I want to say a couple of things about signal transduction in general. So here you can see a general receptor. In fact, on the top you have 3 figures, and on the bottom you have 3 figures. They are 2 different ways many receptors in your body work. They're pretty generalizable. I'm going to talk through the one at the top. The one at the bottom has a lot of similarities. So what you will notice is that this thing at the top, the most famous type of receptor that works this way is something called a receptor tyrosine kinase, or RTK. There's 3 words in its name. It has a domain outside of the cell that is able to bind to the ligand. You could almost think about that as the receptor part. There was 3 words, receptor tyrosine kinase. It's the receptor word of those 3 words. Very frequently, you'll have a receptor that is in multiple parts. Those 2 parts will diffuse through the cell membrane away from each other. They won't always be together. But when ligand comes around, they will bind to ligand, and that will induce proximity of those receptor subunits. So here you can see they're far apart. Once the ligand binds, this brings the binding domains outside of the cell close together. It's called the induced proximity model of signaling. When we bring the outside parts of those proteins together, the parts that are outside of the cell, that by definition pushes the ones inside of the cell together too. The whole protein comes together. And so we get these inside parts of the cell coming together. These inside parts of the cell frequently have a enzymatic domain, a kinase domain. What's a kinase? Yes, Cal? It's an enzyme that phosphorylates stuff. So in fact, we have a kinase that's going to phosphorylate stuff. These lovely pink dots are the phosphorylations that have happened. And those phosphorylations happen on a specific type of amino acid very frequently in many kinds of receptors, which is tyrosine. So that's why these are called receptor tyrosine kinases. They have a receptor part, they have a kinase part, and they have a tyrosine that can get phosphorylated. So when they bind together, they can phosphorylate one another, and that can lead to further signaling. Sometimes the kinase is a separate protein that just travels with the receptor. Sometimes it's just a domain of that same protein. But the big important things here are we need something that's going to bind to ligand, the receptor part. We need a kinase to do the phosphorylating, and we need something to get phosphorylated, typically the tyrosine. So if you remember the name of receptor tyrosine kinase, you remember all three parts. Now I want you to look at the B cell receptor. This is a great view of the B cell receptor from your textbook. If you look at the B cell receptor in this view from your textbook, is there anything that you notice? Yes, Marina? It doesn't go very far into the cytoplasm. It basically stops as soon as it goes through the membrane. Why does that matter? Yes, Amelia?
It's going to be really hard to get any stable transduction if you can't get inside of the cell.
Right. So if we think back to the model of a receptor tyrosine kinase, what parts does this have similarity to? It's a receptor. It is, in fact, incredibly specific for a ligand. It goes through this whole VDJ business to bind to ligand and be an awesome receptor. Does it have a tyrosine inside of the membrane to be phosphorylated? Nope. It basically stops right after the membrane. There's not enough room for a tyrosine. Does it have the enzymatic kinase? Nope. So even though this receptor is great in that it goes through this whole VDJ process to be super specific, doesn't even have all the parts it needs in order to signal. So the B cell receptor works along with some partner proteins. So the B cell receptor gets by with a little help from its friends. There are a lot of different partner proteins that we could talk about. I'm going to mention two here by name, and on a later slide I'm just going to say, and others. So really what I want you to notice here are the two that I'm mentioning by name. The B cell receptor travels along with two other proteins called Ig alpha and Ig beta. They're also known as CD79A and CD79B. And these proteins don't have much of an extracellular domain, but they have a really long cytoplasmic domain. That cytoplasmic domain contains a specific region known as an ITAM. And we're going to see lots of ITAMs this semester. ITAM, like everything else in immunology, is an acronym. And so if we know what it stands for, it will help us out a lot. An ITAM is an immunotyrosine-based activation motif. The really important things as a part of this acronym are the T is for tyrosine. That's the amino acid that gets phosphorylated a lot. So this is providing us a place to phosphorylate. The other thing that's important in this acronym is that the A stands for activation. And so the signaling that happens here is turning on kind of signaling. Later in the semester, we're going to see something called an ITIM. And the I is going to be for inhibitory. And so it's going to be the same thing but turning off kind of signaling. So we have an ITAM, we're turning the signaling on. So our B cell receptor is going to actually work with this co-receptor. There are some really interesting ways that people have shown that these actually do diffuse together in the membrane. They don't separate and diffuse separately. It's actually somebody 2 years ahead of me in my grad school program that showed that. We were very in awe of him. And so we know that these do diffuse together. And that B cell receptor also will have a kinase that's traveling with it. It's probably traveling with some other types of accessory proteins that I'm not going to deal with now. But what I want you to know is, yes, there is a kinase. And should multiple B cell receptors be brought close to each other by binding to antigen, then we will have that kinase phosphorylate the ITAMs, and we'll get our signaling. So it's that same kind of induced proximity model of signaling. A lot of events happen downstream of this signaling. Notice that all of these events lead to things like changes in patterns of gene expression, differentiation, activation, and other changes in the cell. You can see a large number of proteins that are involved in the signaling here. Good news number one, I'm not going to go through them today. Here's the reason why I'm not going to go through them. Later in the semester, we're going to talk about T cells and T cell activation. And the pattern looks very similar. So in fact, we're going to go through all the details of T cell signaling later on. And I'm going to put this back up when we go through T cell signaling at the end. And I'm going to say, does this look familiar? And you're going to be like, it's the same thing. So that's why I really only need to do it once this semester. I don't need to go through the exact same thing twice. And I'm going to go through it with T cells because I like T cells. And it's a better part of the semester for us. Other proteins are probably going to participate as well. They are involved in things like recruiting that kinase. So all of these things are going to happen, are going to be important for helping our B cell to signal. These signals are really important in helping our B cell do its development in the bone marrow and go from being a hematopoietic stem cell to first a lymphoid progenitor and then eventually to a B cell at the end. There are a bunch of steps where our hematopoietic stem cell first commits to becoming a B cell. We're not going to focus on those steps. But those steps involve turning on a lot of specific transcription factors. They involve the cell receiving a number of different signals like cytokine signals or others. Your textbook has this nice figure that shows things like which microRNAs are involved and things like that. So there are a number of different signals between that initial hematopoietic stem cell and this cell that is going to become a B cell. And both of these figures from your textbook kind of talk through how we go from the hematopoietic stem cell to this cell that's getting ready to be a B cell. We're going to really start with this cell that's already committed to becoming a B cell. This cell has a specific name. And this specific name of this cell is the pro-B cell. With each of the types of cells we see in B cell development, there are going to be a few specific things that we're going to notice about those cells. And the first of those things we're going to notice about the pro-B cell. So if we look at this picture of the pro-B cell, or in fact any of these pictures of the pro-B cell, do any of them show a B cell receptor on that pro-B cell? No. In no case do you see a B cell receptor in any of these pictures on the pro-B cell. To be more specific, I'm going to tell you that this cell has no heavy chain and it has no light chain. Really, that means it has no B cell receptor at all. And so our pro-B cell is going to do a specific function. There's a specific thing that pro-B cells do. Really, we see two big things that are happening with pro-B cells. And so sometimes people divide up our pro-B cells based on which of those two events they're doing. I'm going to call them both pro-B cells, but you can see here your book talking about early pro-B versus late pro-B, where they're doing these different events. The first event that is done by a pro-B cell is we rearrange the D region of the heavy chain to the J region of the heavy chain. So the first thing that this pro-B cell does is put this VDJ recombination of the D segment of a heavy chain with the J segment of a heavy chain. The second thing that this pro-B cell does is that it recombines the V segment of a heavy chain to the DJ that it has just made. So step one, you do D to J. Step 1, you do V to DJ. And so that's what is being described here as early versus late pro-B. So here you can see early has a DJ, late has a V to DJ. That's also shown up here. The early pro-B is doing D to J. The late pro-B is doing V to DJ of the heavy chain. So once the pro-B cell has done this function, there's going to be a test to make sure that the function has gone correctly. And this test is related to the pro-B cell going to become the next type of B cell. So we have this first checkpoint that happens. The first checkpoint is really, I would really think of it as being the first thing that the next kind of cell does. Because once we've done D to J and V to DJ of the heavy chain, what do we theoretically have? Yes, Molly. Do you have a whole BCR? No. Yeah. A heavy chain. You ideally have made a heavy chain at this point. And so the next thing that the cell is going to do is test the heavy chain. Because why would you go on and make a light chain if you made a bad heavy chain? That seems pointless. So the next thing that's going to happen is that our cell is going to actually test its heavy chain. The way that it tests its heavy chain is that it is going to put the heavy chain on the surface of the cell and see if it gets a signal. I'm going to skip ahead for just a second. You can see that here. So we're going to now put that heavy chain on the surface of the cell and see if it works. If it doesn't work, then we clearly failed. But if it does work, then we're going to put this receptor on the surface of the cell, and the cell is going to get a little pat on the head. It's going to get a signal through this heavy chain that says, you did a good job. You can keep going. Once the cell puts this heavy chain on the surface, the cell becomes known as a pre-B cell. And so I would say that first step of testing is really the first thing that's happening to our pre-B cell. A pre-B cell now has heavy chain on its surface. But there's a thing that I kind of went past here. When we talked about the structure of an antibody or of a B cell receptor, that structure contained both a heavy chain and a light chain. And in order to fold correctly, you really need a heavy chain and a light chain. But we haven't made a light chain yet. So how does the heavy chain fold correctly when it goes on the surface of the cell? The answer is that this pre-B cell is not putting the heavy chain alone on the surface of the cell. The pre-B cell is actually putting that heavy chain on the surface along with two other proteins that are known as surrogate light chains. You can think of them as like the fake light chains or the sub-light chains until you have a light chain made. So we're going to have these substitute surrogate light chains that are made in order to help put that heavy chain on the surface. The two proteins are known as V pre-B and lambda 5. And you can see them in this figure. So V pre-B and lambda 5 come together and bind to that heavy chain. And when you have a heavy chain plus surrogate light chain, you refer to that as the pre-BCR. Because it's not a real BCR yet. We don't have a real light chain. We have this fake light chain along with our heavy chain. And so we can say that the pre-B cell has heavy chain plus surrogate light chain on its surface. Another way you could phrase that is you could just say the pre-B cell has a pre-BCR on its surface. And that is, in fact, the definition of a pre-B cell. A pre-B cell is a B cell with a pre-BCR. So here you can see our pro-B cell has no heavy chains and no light chain on the surface. but our pre-B cell has a pre-BCR with a heavy chain and our surrogate light chain. And you can also see this here, where now we have the VDJ, we've got a pre-BCR on the surface. If you think about this cell, there are a few things that are going to need to happen. All of them are going to happen as the result of a signal. We want the cell to know it did a good job. It made a heavy chain correctly. It didn't screw up VDJ recombination and make an inappropriate heavy chain. And so what we want is we want the cell to be able to get a signal. We want the cell to be able to get its pat on the head to say, okay, you can keep going. Remember, I told you these cells are very needy. This has been a little bit of confusion in the field for a while because remember I told you we're in the antigen independent phase. So how the heck do we get a signal if we're in the antigen independent phase and we don't even have a real receptor yet? We have like a halfway made receptor. What we think happens is that the surrogate light chain has these weird little loops on the end. You can see V pre B and lambda 5 with their loops here. And those loops from one BCR are able to bind to another neighboring BCR, and BCRs that are actually present on the surface as proteins basically are automatically pulled together, and we get signaling that way. So we think that this is how the pre-B cell receptor will signal, is that the surrogate light chains will pull adjacent B cell receptors together in order to force that signaling, to force the induced proximity that I showed you before, usually happening because of antigen. So now let's think about this pre-B cell, because the pre-B cell has a lot of things that it needs to do. So we've just gone through a whole bunch of effort. We've done a D to J and a V to DJ to make a cell with a proper heavy chain. And we are getting a signal through the pre-B cell receptor, so apparently we did it right. What types of signals do you think the B cell is going to receive at this point? What is the B cell going to want to do? What would we hope the B cell does? Yeah, Molly? Okay, eventually it needs to make a light chain. That's not going to be its first event, but make a light chain is going to be on the list of things that a pre-B cell does. Absolutely. What else does it need to do? Yeah, Leo. So it needs to do recombination again to make the light chain. So that is definitely going to be an event it does. But we got some other things it needs to do. Remember I told you the cell is super needy. So what does it need first? A pat on the head to tell it what? Good job, keep going. Don't die. Don't die. So the first thing we get is that cell actually gets a survival signal. And so this is just two different versions of it. The first thing that happens is that cell gets a survival signal. It's told to survive and not die because of signaling through this pre-B cell receptor. There's going to be a few other things, obviously, as is shown here. We did a good job making a heavy chain. The next thing we want to do is we want that cell to divide and make more of itself. Because in theory, if we have a bunch of copies of that cell, each progeny could make a different light chain, and we could have a whole bunch of different antibodies made. So if that cell divides, then we have more chances for diversity. We've got one good heavy chain, let's divide so we can have lots of different light chains paired with that heavy chain. So the next thing we see is we see a signal for proliferation. We see a signal to push this cell into cell division. First, we're going to see another type of, some other inhibitory signals before we see some other types of positive signals. So one of the things that we see at the beginning with our pre-B cell is actually that pre-B cell stops making RAG for a while. The reason why this cell stops making RAG is because it's going to proliferate. It's going to go through the cell cycle. And RAG, our DNA breaking enzyme, is a pretty dangerous thing to have around when you go through cell cycle. And so at any step where our developing lymphocyte is going to go through cell cycle, any step where we're going to see a lot of proliferation, we stop making RAG. So that we have a period of time where we can divide and have our DNA be copied safely without getting cut. So we're going to turn off RAG. We're also going to turn off production of surrogate light chain. We're not going to make any more V pre B or lambda 5, which makes sense. We just used it. We have done the one step we need with that surrogate light chain. Ideally, the next step is we're going to make light chain. So we're not going to need the surrogate light chain anymore. We're going to make a real light chain now. So we can turn off production of surrogate light chains. There's one other thing that happens before we get to recombination and making the light chain. And this is a thing that people struggle with a little bit. Officially, the thing that we're going to talk about is called allelic exclusion. If you look at the word allelic, what word does it look like? Allele. So what do you think this has something to do with? Alleles, chromosomes, genes, that kind of stuff. What's exclusion mean? Keep out. So there's something about keeping out genes going on here. And that is in fact true. To really understand allelic exclusion, we have to come back to this sort of figure. We just did a process where we put a D with a J, and then we put a V with a DJ on chromosome 14 with the heavy chain. We have not yet done the light chain. Make sense, yes? How many copies of chromosome 14 do you have in all your cells? 2. What would happen if both of your chromosome 14s rearranged in your developing B cell? If your mom's chromosome, if you made a mom heavy chain on mom's chromosome 14 and a dad heavy chain on dad's chromosome 14, what would happen? Anybody? Okay, there's nothing left over, but there's another bigger problem. I always want colored chalk on the allelic exclusion day, and I never remember it. So we've got our B cell. Our B cell is going to be giant for its moment. So this B cell could make mom's heavy chain, could make dad's heavy chain, and it could actually make mom's kappa, could make mom's dad's kappa, could make mom's lambda, that was just not going to go right at all. And it could make dad's lambda. Ooh, that's supposed to be dotted. And this is assuming that all B cell receptors are symmetric, which if this actually happened would not be the case. And so you could also have like 4 different proteins bound together. And so what you would notice is that this B cell in this view has 8 different B cell receptors and can make 8 different kinds of antibodies. So what happens if this one responds to peanuts and this one responds to flu? Suddenly, if you kill this cell in deletion, you've killed the peanut allergy cell, hooray, but you also killed the flu cell. So in fact, each developing B cell and each developing T cell has only one specificity. It makes only one kind of heavy chain and only one kind of light chain. And allelic exclusion is the big piece to this. When we have a successful heavy chain that is produced, we shut off further recombination of the heavy chain. So let's imagine, and so first let's just look at this other figure of allelic exclusion. So here you can see the potential problem. With what actually happens, our B cell always has multiple copies of one B cell receptor that are identical. This is good because then you can't have a cell that's both kind of self-reactive and non-self-reactive. The cell has one thing it's reactive against. But also you can get good cross-linking because you can have many B cell receptors binding to the same antigen and you can get a nice strong signal. With this B cell, we're confused. Are we self-reactive, are we not? Or what the heck are we reactive to? And we can't get a very strong signal because we can't cross-link multiple of these receptors together. And so we need to stop this from happening. So we're going to inhibit further recombination. The way that this really works, you can think about it, is that you're going to start doing recombination. RAG doesn't know which one is the mom chromosome and which one is the dad chromosome. RAG is going to start doing recombination. One chromosome is going to finish first. It's just how time works. One of them finishes before the other one. Probably they both make it through D to J, and then one chromosome makes it through V to D to J first. And as soon as that is done, the heavy chain goes up on the surface. And as soon as that heavy chain goes up on the surface, it stops the other one from being able to do its V to D to J. The signal that comes through this receptor inhibits further ability to recombine at the heavy chain locus. So one chromosome gets to complete VDJ, the other one is excluded. Does this make sense? Yes, Cal. You do want a lot of diversity, but you want each cell to be have only one specificity. And so the idea here is that each cell is going to get one specificity this way. And so it's possible that the neighboring cell, a different chromosome won. And so the idea here is you're doing this in so many cells that you get the diversity. So this sort of is a little bit of a best of both worlds kind of thing. Yeah, Molly.
This might be repetitive.
That's okay.
Is there a statistical equivalence between which chromosome gets selected? Like, do they have an equal chance? Does one always get selected?
To my knowledge, at this step, there is no difference. They get picked equally often, or they finish equally often first. Okay? All right, so eventually we're going to make light chain. So the next thing that's going to happen here is that we're going to have this cell make light chain. But we've been making an assumption thus far in our pre-B cell. We've been making an assumption that the heavy chain we made worked. All of these signals that we get are signals that we get through a heavy chain that worked. We didn't talk about what if the heavy chain didn't work? What if it failed? And so there's also something that happens if the heavy chain fails. What we're going to do is we're generally going to do our D to J rearrangement on both chromosomes, as I mentioned before. We'll then see V to DJ chromosome on the recombination on the first chromosome, and we'll try to get a signal through that receptor. If we get a signal, we get the pat on the head to survive, we stop heavy chain recombination, we proliferate, we stop making surrogate light chains, all the things I told you about. But if that heavy chain has failed, if the heavy chain we made is a bad heavy chain, there is no signal to stop recombination. And so now the second chromosome finishes recombining. We get a second chance to try to make a good heavy chain. We have those two chromosomes, so we get two tries. So if the first one fails, see here's fails with the negative sign and the red arrow because it's sad. Then we try the second chromosome. If the second chromosome makes a heavy chain, then in fact that B cell is going to survive. If the second chromosome also fails, then the cell dies. And you can see half of the cells fail with both chromosomes and are just out of luck. You can see this from your textbook here as well. Our B cell is first going to do D to J recombination. It will then try to do V to DJ on the first chromosome. It might do a good job and make a pre-BCR. If it fails, it'll try its second chromosome, might do a good job, make a pre-BCR, might fail, and then die. Yes, Emilio?
And the chance that it goes to use the second chromosome, what happens to the first heavy chain? Does that interact with anything? Or is it just
I don't know of anything that it interacts with, but let me come back to that in just a second, okay? So the question that I wanted to ask here is, why do you think so many of these are failing? Realize that I said 50% fail on both chromosomes. So you can imagine like 25 make it on chromosome 1, 25 make it on chromosome 2, 50% fail altogether. That's a pretty high failure rate. So my question for you is what do you think is happening that makes all of these VDJ recombinations fail? There are a lot of reasons, but there's one really big one that I want to point out. So, oh yeah, Marina?
It's a guess.
Okay.
But when they're doing the VDJ, when you're adding the nucleotides, if it's off.
Yeah. So remember how we added those P and N nucleotides? We opened the hairpin and added random nucleotides, and then we added some other random nucleotides with TDT. What happens if you added a multiple of that wasn't 3? What happens if you added 5 base pairs total? Yes, Molly? You have a reading frame shift, and you're not going to get a full protein. You're going to introduce a stop codon and get a truncated protein. So only if those additions of P and N nucleotides work out in a multiple of 3 are you really going to be able to get a functional protein. So most likely, Emilio, the failed heavy chain is a truncated, shortened heavy chain that had an early stop codon, so it's not going to be a full protein. So that's the big reason for failure, though we can imagine other reasons for failure as well. Yep, Jordan.
Would that then be degraded? Would it
It would the protein would probably get degraded by the unfolded protein response because it couldn't be folded correctly. That's really all that we got on that. So one thing I hope you realize here, like with other places with immunology, efficiency, not a word we worry about with immunology. We are making huge numbers of cells, and many of them are going to die and make non-productive rearrangements and not make it through this process. Once our pre-B cell has done all of these things, once our pre-B cell has survived and proliferated and stopped making heavy chain and done allelic exclusion and had all of this work out, then it finally does its last job. And what do you think is the last job of the pre-B cell? What do you think, Malik? Make the light chain. It finally gets to make the light chain. And so now this cell, which had previously turned off RAG, is going to turn on RAG and start doing recombination again. So our pre-B cell will have its pre-BCR on the surface, and it will now, after doing all sorts of other jobs, after proliferating, after surviving, after allelic exclusion, after turn off, surrogate light chain, now finally it's going to do the light chain V to the light chain J. We're finally going to get to do recombination of our light chain. And so you can see here, finally, VL to JL rearrangement happens. So, you had to guess. When we finish light chain rearrangement, what do you guess is going to happen next? Cal. We're going to test it now. So it's now the same process. And how do you think we test it? What do you think we first have to do to test it? Yeah, Emilio?
Assemble a functional BCR.
Yeah, you got to put it on the surface. You got to assemble that B cell receptor on the surface. Previously, you had a pre-BCR on the surface. Now we have to have the actual BCR with the heavy chain and the light chain. A pre-B cell is defined as a B cell with a BCR on its surface. So when you now put up this new heavy chain and light chain, this new BCR to test, you also get promoted to having a new name. Because you no longer have a pre-BCR on your surface. You now have an actual BCR on your surface. And so at this point, you can now be called an immature B cell. You have a heavy chain plus a light chain, otherwise known as an actual BCR, on your surface as an immature B cell. So the immature B cell is largely going to have turned off RAG. It's really going to have turned off a lot of our recombination enzymes. So for example, it doesn't have TDT. It doesn't need that IL-7 receptor anymore because it has a receptor now where it can get signals. It has its own receptor for a pat on the head. It doesn't need that survival one anymore. And it will start making some other B cell specific proteins. That first thing that's going to be made is going to be IgM. That it will be on the surface of our cell. So we're making a B cell receptor that's using the IgM constant region, which was the first one, the most 5' constant region at this point. So these are immature B cells. However, these cells are still in the bone marrow. These cells have not yet left to go into the periphery because these cells still need to do some testing. First, we need to test the light chain. Once we test the light chain, we also need to actually decide, is this a self-reactive cell? We need to look for self-reactivity. So we still have some work that this cell has to do in the bone marrow. Only when it's done with those steps will it be able to leave the bone marrow and finally be a mature B cell. So the first thing we want to do is test the light chain, and we're going to be testing to see if we can get a signal through the light chain. Testing the light chain is pretty similar to testing the heavy chain. There are a few kinds of signals that our cell gets when it tests the light chain. So what kinds of signals do you think we get? Sorry. What kinds of signals do you think we get? What do you think, Manny? You get a survival signal, just like you got before. What other kind of signals do you think you get? You get a proliferation signal, just like you got before. What other kind of signals do you think you get? Yes, Malik? So you're going to get allelic exclusion again. With the light chain, we've got mom and dad's chromosome, and we've got a kappa light chain and a lambda light chain. So there's 4 different ones that we've got to allelically exclude, or 3 others that we have to allelically exclude. We're also going to turn off RAG at this point. The immature B cell has one good thing going for it. With our pre-B cell, who's made a heavy chain, we got 2 tries. And if we didn't get a good heavy chain in 2 tries, the cell died. These green cells with a mu in them are our cells that were successful in making a heavy chain. And once they go to make a light chain, they first try to do rearrangement of kappa. We don't understand any functional difference between the kappa light chain and the lambda light chain, but we do know that kappa seems to be tried first in VDJ recombination. So we try recombination at the kappa, at one of the kappa loci. If it works, hooray, the cell lives and gets to be an immature B cell. If it fails, then in fact, we try the other kappa. If it works, hooray, we allelically exclude the rest, we get to survive and be an immature B cell. If it fails, then the B cell gets to try the first lambda. If the first lambda works, hooray, the cell lives. If it doesn't work, the cell tries again and gets to try a second lambda. If it works, hooray, the cell lives. If not, now the cell is going to undergo apoptosis. So with the light chain, the cell gets 4 tries to make an appropriate light chain and be able to actually make a proper B cell. Once the cell has done this rearrangement, it shuts off RAG, and it shuts off RAG for the rest of its life. So the B cell is largely done with RAG. You're going to see RAG is going to come up in one place with the testing. That's happening like right at this same stage. But otherwise, we're done with RAG. With the T cell, it's totally done at this point. And so now what we have to do is we've got to test those B cells. And we're going to test those B cells for a specific thing. This now is where we're going to test for self-reactivity and potentially delete any B cells that are self-reactive. So we have just done this phase of diversity, and the next step for our B cell is the testing that allows us in this figure to get rid of number 1 and number 4 because they're self-reactive. And so now we have to delete self-reactive B cells. So you've seen 2 pieces of the clonal selection hypothesis here. First, you notice that the immature B cells all bear Ig receptors on their surface, and all the receptors on a single B cell have identical specificity for antigen. That's a fancy way of saying allelic exclusion happens, and it's important. We are now at the point of the one that's at the bottom, which is that B cells with receptors for self-antigens are going to be deleted during development. And that's largely the step that we're going to be seeing on Monday. The one piece I will say is that this process in general of deletion is frequently referred to as central tolerance. Tolerance is how we get rid of self-reactive cells in the immune system. It's how you get rid of autoreactivity. If you have an autoimmune disease, you have a failure of tolerance. It's how you learn to tolerate yourself. There are different ways tolerance can happen. They can either happen during development or later in the immune response. And so tolerance mechanisms that occur during development are known as the central tolerance mechanisms. So what you're seeing when we see deletion next time is really the central tolerance mechanism.